Of the session is Veronique Bolduc from NINDS, and she's going to present her work on collagen 6 mutations, looking at allele-specific targeting of pathogenic variants acting as dominant negative actions, and then looking at that as a potential therapy. Okay, so Veronique, are you ready to go?
Yes, thank you. Thanks for the invitation. I'm happy to report on our data. So one feature that nucleic acid therapeutics have in common is that they function through base pairing. So small interfering RNAs, antisense oligonucleotides, and guide RNAs can be designed, rationally designed for very specific RNA or DNA targets. And we can use this feature to our advantage in the case of dominant variants that encode gain-of-function or dominant negative protein isoforms. So by designing an antisense drug that's specific for the variant, we can take away the protein isoform that's mutant and rescue the phenotype, as long as the dosage is not an issue. So the collagen 6 related disorders are good cases to test those approaches. In brief, these are a complex group of disorders. They manifest with a mix of muscle weakness and joint contractures with various degrees of severity. They are rare but found worldwide. And they are caused by pathogenic variants in either of these three complex genes, collagen 6 alpha 1, alpha 2, or alpha 3. And these variants can be inherited as recessive, dominant, or can occur de novo. And both mechanisms can lead to mild or severe phenotype depending on the consequence of the mutations. So I'll give just two examples of severe variants associated with severe phenotype. Recessive null variants that lead to complete absence of collagen 6 in the tissue are severe, as are dominant negative variants that lead to complete mislocalization of collagen 6 in the tissue. So while this picture seems complex, there are a few key points that I'd like to draw your attention to. First, obviously, these disorders have a clear genetic etiology. The dominant, de novo dominant negative variants are far more common in mixed populations such as the US. And not only that, they appear in three categories. So they're either glycine substitution of the GlyXY repeats, in frame exon skips, or in frame pseudoexon insertion. And they cluster at one end of the triple helical domains. So we can imagine an approach for each of these classes and hope to be able to expand this approach to all variants within the same class. And finally, carriers of one null variant are asymptomatic, such as the parents of recessive patients, which means that these genes are not haploinsufficient, which means that we can use allele-specific targeting for these genes. So just a brief schematics to visualize collagen 6 production. Those three genes encode three polypeptides, alpha chains, that assemble with one another through their respective triple helical domain to form a single monomer. The dimerize, tetramerase, and tetramers are secreted to form this collagen 6 microfibrillar meshwork. Now, in the lab, we routinely use fibroblasts cultured from skin biopsies, as these cells express the three genes and they lay down a matrix at the cell surface that we can image and quantify with immunofluorescence. Some dominant negative mutations, when they are assembly competent, they can have a very strong dominant negative effects, such as the majority of tetramers contain at least one mutant chain, which has an impact on the ability of these to form a matrix. So, of course, our goal is to suppress the production of those dominant negative isoforms. So here's a table summarizing our approach for each mutation class, variant class. I'll be presenting allele-specific silencing with siRNAs for one exon skip variant, as well as one glycine substitution. and allele-specific gene editing for another glycine substitution. I do not have time to discuss our exon skipping approach for a pseudoexon, but I invite you to abstract number 2, and this poster can be found in the category Other, and with keywords such as skipping. And there's also Carson's talk tomorrow at 1:30 p.m., I believe, and he'll be touching on this topic as well. So here, we started this work by targeting this common exon 16 skip variant in the collagen 6-alpha-3 gene, and we used a unique exon 15-17 junction to design siRNAs specific for the mutant transcripts. And we quickly found 2 siRNA that, when transfected in patient fibroblasts, were highly efficient at knocking down the mutant transcript without affecting the expression of the wild-type allele. So this was determined by quantitative PCR. One effect of the dominant negative isoform is impaired matrix deposition in culture, but upon allele-specific silencing, the matrix deposition was increased. Now, with these, in these culture conditions, we estimated that we could reach down to 75% of knockdown of the mutant allele, and this was sufficient to significantly increase the matrix deposition. So without necessarily reaching 100% knockdown, we improved the matrix. So now this was pretty straightforward since exon skip offer good targets for allele discrimination. What about glycine substitutions? Such as the glycine 293 arginine variant that's caused by a single nucleotide change in the COL6A1 gene. And this work I'm presenting was initiated by Apurva, is now led by Astrid in our lab. So here what we did is we designed reporter construct for each of the allele in order to be able to look at the effect of each siRNA against each of these alleles. And our initial screen of siRNAs, we found one that was highly efficient at knocking down the mutant allele, but it was also knocking down the wild-type allele. So to increase the allele specificity here, we did this design modification where we introduced mismatches going towards the seed sequence, such that each of these siRNA now have 2 mismatches compared to the wild-type allele. And again, testing those siRNAs against our reporter construct, we were able to identify a few that were still efficient against the mutant allele, but that would rescue the wild-type, would preserve the wild-type expression. So these, here is the quantification of these plots. The mutant allele is suppressed, but the wild-type allele is preserved. And we can see this again with immunofluorescence. So with just a small design modification, we could drastically increase allele specificity. So we're currently testing those siRNAs in patient fibroblasts, looking at allele-specific expression of the 2 alleles and also matrix production in culture. So now for a more permanent solution, we ask whether we can use gene editing in an allele-specific manner. So here we intended to inactivate one variant by the random introduction of frame shift edits by non-homologous end joining. So in the case here, we were not only looking at the capacity of guide RNAs to discriminate between the alleles, but we also investigated the repair outcomes introduced by non-homologous end joining. This work was initiated by a former summer student in our lab, Catherine, and we received help from Brian and Eric for the bioinformatic analysis. So here our model was this glycine 290 arginine substitution because the variant sits right next to a PAM sequence. So we designed what guide RNA that was specific for the variant allele. We cloned this guide RNA into a Cas9 GFP plasmid, transfected patient cells, and used cell sorting to enrich for GFP positive fibroblasts. And from there, we used targeted resequencing to profile the edits that were introduced at both alleles. And so we could find that after guide RNA treatment, more than 90% of the mutant allele were edited, showing that this guide RNA is quite efficient at this allele. However, close to 50% of the wild-type alleles were also edited. The most common edits found were one base pair deletions at the mutant allele, but also at the wild-type allele. So to look whether these occur at the coding sequence to effectively promote frame shifting, we looked at the reads, and indeed the vast majority of edits were one base pair cytosine deletion in the coding sequence. So overall, the majority of the edits introduced were causing frame shift, but these were also present in the wild-type reads. So currently, we're testing whether we can improve the allele specificity of guide RNA the same way we can do with siRNA. But in the meantime, we still tested these cells in culture for matrix production. And so basically after guide RNA transfection, sorting, we plated the cells as a mixed population, and we did find increased matrix deposition, even though the allele specificity was not complete. So which means that there are sufficient number of cells here that were correctly edited only at the mutant allele, and that the collagen-6 secreted by those cells could produce a matrix. So this is highly encouraging, even though allele specificity is not reached completely yet. So in conclusions, well, of course, I wanted to make the point that allele-specific targeting is appropriate for dominant variants in haplosufficient genes, such as the collagen-6 genes. They can be designed for any antisense-based molecule. I provided example for siRNA or guide RNA, but you can find examples in the literature for those two and also for antisense oligo. Even if allele specificity is not optimal at first, it can be optimized with design modifications or by changing the chemical, the modifications of the bases, which is done by other groups. And the balance between potency and selectivity is something that might be have to be determined for each design, each disease scenario. But in the case of the collagen-6, our current hypothesis is that any amount of reduction of the dominant negative product will provide benefit. And finally, I just want to leave you very quickly on these other additional ideas for allele-specific strategies, how to utilize allele specificity. So what I've shown today were molecules designed specifically for a variant, but we can also use a design where we use a SNP that's in proximity of the variant. So if we design a molecule for each allele of the SNP, as long as the patient is heterozygote and we can phase the SNP with the causative variant, then we can use one of those two molecules to knock down specifically the transcript that's related to the variant. So with just a few molecules, we could potentially cast a much wider net of variants. I spoke about gene downregulation, but of course this can be used for gene upregulation if targeting promoters or allele-specific steric blocking. So finally, I want to thank everyone in the Bonneman lab in particular, those individuals here who contributed directly to the work I presented here, are bioinformatics and cell sorting facilities.